The nucleus of each cell in your body contains DNA. DNA is responsible for storing and transmitting genetic information from one generation to another. The monomers of DNA are called nucleotides, which make up the structure of DNA. Each nucleotide contains one phosphate group, one 5-carbon sugar, and one nitrogenous base. The 4 nitrogenous bases are adenine, thymine, guanine, and cytosine, in which adenine pairs with thymine and guanine pairs with cytosine. Chargaff's rule, named after chemist Urban Chargaff, states that in DNA there are equal amounts of adenine as there is thymine, and equal amounts of guanine as there is cytosine. Rosalind Franklin was the first scientist to discover the structure of DNA. He used an X-ray to spin DNA and found out about its spiral structure. The double helix model was made by Watson and Crick. This model explained how DNA could replicate itself and how hydrogen bonds between bases hold the 2 strands of DNA.
DNA replication ensures that each new cell that is made has a complete set of genetic information. In order to make a new cell, the old cell has to copy the DNA before they divide. As a result, double-stranded chromosomes are produced, each with one new strand and an old strand. The first step in the process of DNA replication is for a weak hydrogen bond to break and allow the DNA to zip apart. The 2 strands then have to unwind. After this, a DNA enzyme called polymerase bonds to the corresponding nucleotides.
RNA, aka ribonucleic acid, has the main job of making proteins. There are 3 types of RNA. mRNA, which is the messenger that carries a copy of the instructions. tRNA, which transfers amino acids to a specific code from mRNA. And rRNA are molecules that translate mRNA molecules to produce proteins. There are also a few differences between RNA and DNA. One of them includes RNA being one-stranded, while DNA being two-stranded. Another difference is adenine is paired up with thymine in DNA, while in RNA, adenine is paired up with uracil. DNA is located inside the nucleus, while RNA is located outside the nucleus throughout the cytoplasm. RNA is like a copy of a blueprint. DNA is the original copy. If the original copy is lost, then you cannot make more copies without it. Transcription happens in the nucleus. Its job is the process of copying DNA to RNA. DNA copies itself to RNA through codes called codons. Codons are every code that is read in 3 letters, which are specific for an amino acid. If the wrong amino acid is used, malfunctions like mutation in breast cancer can occur.
Translation is the process of translating codes from mRNA to make a polypeptide chain, which is a protein. And unlike transcription, translation happens in the ribosomes outside of the nucleus. Once the mRNA leaves the nucleus, the ribosome reads its instructions to make polypeptide chains. tRNA is brought with an amino acid for each codon on the mRNA chain. The anticodon end of the tRNA connects to the codon on the mRNA. The tRNA continues to go to other mRNAs and the polypeptide chain grows until a stop codon releases it.